So anyway, on to MacConkey Agar. This is sometimes also called Mac. Let me see that up here. And MacConkey Agar is now a gram-negative agar. This particular type of agar is selective and differential. And this one is selective for gram-negatives, which means it kills gram-positive organisms and allows our gram-negative species of bacteria to grow. Now, how does it do this? It works on the exact same concept as our mannitol salt agar. Something inside of it is going to kill gram-positive agar, or gram-positive species, and that something, whatever it happens to be, the selective agent, does not impact gram-negatives. Gram-negative bacteria don't care. They will be able to survive in it. Now, in this case, what is that selective agent? Well, in this case, for MacConkey Agar, there are two of them. It is crystal violet. Crystal violet, the same thing that's used for gram stains, the purple color, as well as bile salts. Now, bile salts are produced in your gallbladder, stored in the liver, or sorry, produced in your liver, stored in the gallbladder, and they are used in your digestion to help emulsify fats. Now, bacteria that live in your intestines, E. coli being a gram-negative perfect example of this, will be coming in contact with large amounts of bile. Bile is very, very, very corrosive. So to be able to survive in the intestines, you have to be able to survive in the presence of bile. So bile salts are going to be added to this media. Gram-positive organisms, they don't live in your intestines. There's no bile on your skin, so they cannot survive in the presence of bile. They will die in the presence of bile. So the bile will kill them. On this agar, if you put them on here, the bile will kill them. Now the other one I mentioned is crystal violet. Crystal violet is a very slight, very mild antimicrobial, antiseptic. Some people use it on livestock. It's called gentian purple, and things for cuts. Sometimes they use it as medicine in the 70s and 80s, 60s. You can sometimes find it in a cream form still. Anyway, it contains both crystal violet and bile salts. Both crystal violet and bile salts by themselves are a weak selective agent. There are quite a few gram-positives, Enterococcus being one of them, that's even diagnosed using a bile plate. It contains nothing but bile. But quite a few gram-positives can survive in the presence of bile. Quite a few gram-positives can also survive in the presence of crystal violet, but they can't survive in the presence of both, and that's how that works. If you can survive in the presence of bile salts, the crystal violet will get you. If you can survive in the presence of crystal violets, the bile salts will get you. So very few gram-positives can grow on this. This is used for gram-negatives. You do a gram stain, you see a gram-negative organism on your plate, on your gram stain, and you go to one of these plates. And that's going to tell you something very, very, very important about this particular agar. So now that you know how the selective concept works, let's talk about how the differential concept works. This differential concept works the exact same as our mannitol salt agar, except replace the sugar with mannitol with the sugar lactose. If you can digest the sugar lactose, if you can ferment the sugar lactose, you are going to produce acidic waste byproducts, which in this case are going to react with the pH indicator neutral red, not phenol red as in mannitol salt agar, but it's the exact same concept, acidic versus waste products based on if you can eat sugar or not. So this is a mannitol or a MacConkey Agar plate right up here with nothing on it, just right out of the package or right when you make it. And this is a MacConkey Agar plate from a lactose fermenting organism. This bacteria, you put it on here, and it was able to ferment lactose. Lactose is the sugar that's found in this agar in high concentrations. This particular organism was able to eat that lactose, fermented the lactose, produced large amounts of acidic waste byproducts. That acidic waste byproduct went out into the media, diffused out it from the cell, reacted with that pH indicator neutral red, and turned that neutral red a bright highlighter pink color. So this guy is able to eat the sugar lactose. Now over here you'll see it went from this kind of watermelon purple to kind of a tan. You can see the other color change over here. This particular species of gram-negative is unable to eat the sugar lactose. So what's the default answer? What are you going to eat if you cannot eat the differential agent, the lactose, the sugar? Proteins. So when you eat proteins, you hydrolyze proteins, you produce a basic byproduct, basic waste byproduct. So this particular organism here, whatever species this is, could not eat lactose. It ate the proteins that are found in that media, produced a basic byproduct as a waste. That basic byproduct was released out of the cells, diffused into the agar, came in contact with neutral red, and turned that neutral red into a brown color, coming in contact with that basic pH. So you would look at this particular plate and go, oh, this particular organism is unable to ferment lactose. And that gives you the next step in your diagnostics of what this is. Can it eat lactose or not? So you can see a plate here that someone split down the middle. One half of it with a lactose fermenting bacteria, one half of it without a lactose fermenting bacteria. So the big key here is, can you eat lactose or not? Selectively, the crystal violets and the bile salts are going to kill our gram positives and allow only our gram negatives to grow. The differential agent in this case is lactose, based on, once again, that concept of, can you eat lactose or not? Producing basic or acidic waste byproducts, which will come in contact with the pH indicator, which tells you what you're looking at species-wise, or what next step to take to diagnose this particular bacteria. Now, the big key here for this particular guy is it breaks apart the lactose fermenting bacteria from the non-lactose fermenting bacteria. This is a big key concept in microbiology here. Our lactose fermenting bacteria are in the family Enterobacteriaceae, enteric bacteria. These guys are not. And this is our big key characteristic here. It's going to break apart those two families of bacteria. How do you diagnose the two families? You tell them apart based on their reactions of lactose. Can you ferment lactose or not? And that's how you tell apart the two different families for the most part. This is going to be the first step to breaking those two guys apart. And if you can't grow on this agar, the crystal violet and the bile salts killed you, which means you're probably a gram-positive organism. They were not able to grow. So once again, here's a video for you guys if you want to watch someone else talking about how MacConkey Agar works. So once again, I would expect you guys on your lab exam to be able to, if I give you a picture of a pink agar, is this bacteria species able to ferment the sugar lactose? Is it able to survive in the presence of the differential agent, or of the selective agent, which is crystal violet and bile salts, which in this case, yes, it's MacConkey Agar. You knew it is. I'll tell you that part, by the way, for the exam. I'll tell you what you're looking at. I'll also provide you with a plate of nothing on it. So you don't have to just instantly go, oh, I don't know what that is. I'll tell you what type of plate it is, as well as what they look like to start with. So your job would be to tell me, can this particular species eat the selective agent or ferment it? Can it survive in the presence of the differential agent? Something like that. So it's kind of what an idea to expect.